UN 영어뉴스 Scientists grow whole model of human embryo without sperm or egg. A stem cell derived human embryo model showing blue cells embryo, yellow cells yolk sac, and pink cells placenta. Scientists have grown an entity that closely resembles an early human embryo without using sperm, eggs, or a womb. The Weizmann Institute team say their embryo model, made using stem cells, looks like a textbook example of a real 14-day-old embryo. It even released hormones that turned a pregnancy test positive in the lab. The ambition for embryo models is to provide an ethical way of understanding the earliest moments of our lives. The first weeks after a sperm fertilizes an egg is a period of dramatic change from a collection of indistinct cells to something that eventually becomes recognizable on a baby scan. This crucial time is a major source of miscarriage and birth defects, but poorly understood. It's a black box, and that's not a cliché. Our knowledge is very limited. Prof. Jacob Hanna from the Weizmann Institute of Science tells me, starting material, embryo research is legally, ethically, and technically fraught, but there is now a rapidly developing field mimicking natural embryo development. This research, published in the journal Nature, is described by the Israeli team as the first complete embryo model for mimicking all the key structures that emerge in the early embryo. This is really a textbook image of a human day 14 embryo, Prof. Hanna says, which hasn't been done before. Instead of a sperm and egg, the starting material was naive stem cells, which were reprogrammed to gain the potential to become any type of tissue in the body. Chemicals were then used to coax these stem cells into becoming 4 types of cell found in the earliest stages of the human embryo. Epiblast cells, which become the embryo proper or fetus. Trophoblast cells, which become the placenta. Hypoblast cells, which become the supportive yolk sac. Extra embryonic mesoderm cells. A total of 120 of these cells were mixed in a precise ratio, and then the scientists step back and watch, creating a model of the human embryo. 1. Starting materials, stem cells. 2. Transformed into 4 types of cells. A. Epiblast. 2. B. Hypoblast. C. Extra embryonic mesoderm. D. Trophoblast. 3. 120 cells mixed and grown in shaker. Cells develop. 4. Spontaneously forms embryo-like structure. About 1% of the mixture began the journey of spontaneously assembling themselves into a structure that resembles but is not identical to a human embryo. I give great credit to the cells. You have to bring the right mix and have the right environment, and it just takes off. Prof. Hanna says, that's an amazing phenomenon. The embryo models were allowed to grow and develop until they were comparable to an embryo 14 days after fertilization. In many countries, this is the legal cutoff for normal embryo research. Despite the late-night video call, I can hear the passion as Prof. Hanna gives me a 3D tour of the exquisitely fine architecture of the embryo model. I can see the trophoblast, which would normally become the placenta enveloping the embryo, and it includes the cavities called lacuna that fill with the mother's blood to transfer nutrients to the baby. There is a yolk sac which has some of the roles of the liver and kidneys, and a bilaminar embryonic disc, one of the key hallmarks of this stage of embryo development. Making sense? The hope is embryo models can help scientists explain how different types of cell emerge, witness the earliest steps in building the body's organs, or understand inherited or genetic diseases. Already, this study shows other parts of the embryo will not form unless the early placenta cells can surround it. There is even talk of improving in vitro fertilization IVF success rates by helping to understand why some embryos fail while using the models to test whether medicines are safe during pregnancy. Prof. Robin Lovell-Betz, who researches embryo development at the Francis Crick Institute, tells me these embryo models to look pretty good and to look pretty normal. I think it's good. I think it's done very well. It's all making sense, and I'm pretty impressed with it, he says. But the current 99% failure rate would need to be improved, he adds. It would be hard to understand what was going wrong in miscarriage or infertility if the model failed to assemble itself most of the time. Legally distinct, the work also raises the question of whether embryo development could be mimicked past the 14-day stage. This would not be illegal, even in the UK, as embryo models are legally distinct from embryos. Some will welcome this, but others won't like it, Prof. Lovell-Betz says. And the closer these models come to an actual embryo, the more ethical questions they raise. They are not normal human embryos. They are embryo models, but they are very close to them. So, should you regulate them in the same way as a normal human embryo? Or can you be a bit more relaxed about how they are treated. Prof. Alfonso Martinez Arias from the Department of Experimental and Health Sciences at Pompeu Fabra University said it was a most important piece of research. The work has, for the first time, achieved a faithful construction of the complete structure of a human embryo from stem cells in the lab, thus opening the door for studies of the events that lead to the formation of the human body plan, he said. The researchers stress it would be unethical, illegal, and actually impossible to achieve a pregnancy using these embryo models. Assembling the 120 cells together goes beyond the point an embryo could successfully implant into the lining of the womb.